Now we are going to be talking about genetic information. Specifically, today we're going to be talking about the polymer DNA. So, characteristics of life, you have to have genetic code, you have to have genetic information. That's the G in our egg chart. So, we know that if we are a living organism, plant, animal, bacteria, whatever, we have to have some type of genetic information. DNA is going to reside in the nucleus of a cell. If you zoom in on it, so let's break apart what is actually in the DNA. So we know that DNA is a biomolecule. We know that it is a biomolecule, a molecule of life. It is one of the nucleic acids. CHONP, we got DNA and RNA. The monomer is a nucleotide. The DNA is going to be in the double helix shape. In between each one, you have one side of the ladder. There are 4 different steps that you can have on your ladder. So here's the basic DNA. Let's zoom into this portion right here. We're going to look at this exact portion and basically just blow it up. When we're talking about our monomers and our polymers, we know that one bead is our monomer and many beads is our polymer. So this little circle right here that I've circled in green, this is circling one monomer. You click all these together and you get your string of DNA. Inside the monomer, you are going to have a phosphate connected to a sugar, and then finally connected to what's called a nitrogen base. Each nitrogen base is different. So if I'm trying to look at my DNA and I'm trying to look at someone else's DNA, we may have the same 4 different nitrogen bases. However, the combination is completely different. The combination is what actually codes for the DNA. Again, similar to like computer code, binary, where you have just zeros and ones. You only have 4 letters for DNA, but the ridiculous amount of combinations you can do by having longer length of code or just switched around letters can actually create all the specific traits and pretty much all the instructions for the entire human body, which is surprisingly complex. Let's say that blue is going to be our A. I don't really mind if you just know the letters. However, if on a test or on whatever you're doing, they say, hey, we're talking about adenine, you have to understand that the A in adenine, it's talking about the nitrogen base. So A is adenine. Look at our picture. The blue is always going to bond with the red. So we are going to assign the red T, thymine. Then we look at our purple and our orange. Again, the colors are just randomly assigned. They're not actually blue and red or purple orange. Cytosine is going to represent our C. And then orange, finally, guanine is going to represent our G. So here's the easiest trick. Here's the trick on how to figure out how to combine these. We said A and T go together, and C and G go together. They are always going to pair in the DNA. The little trick to remember is apples in trees and cars in garages. Apples and trees, cars and garages. This is the easiest way to remember it. You're probably not going to think that a car goes in a tree or an apple in a garage. So if you ever forget what bonds apples and trees, cars and garages, this is called base pairing or Chargaff's rule. He basically said that only these specific nitrogen bases can bond to each other. And again, it's not about colors, it's not magnetism, it's more of chemicals bonding together. So let's summarize. We have a phosphate that bonds over here to a sugar, which bonds over here to a nitrogen base. This is our monomer. This is one, even though it's 3 pieces, it seems weird. Even though it's 3 pieces, this is still one Mardi Gras bead or one Lego block. This is our monomer. you'll notice that it's a double helix. It's a twisted ladder. So this is one side. So right here, phosphate and a sugar, that makes up the backbone or the side of the ladder that you would like grab with your hands. The nitrogen bases make up the rung, which is what you would put your feet on. However, there's one rung on one side and the other rung kind of on the other side, and they connect together. That's where the two colors connect. So let's say that we have a nitrogen base over here as well. This is the other side of our DNA. In between the nitrogen bases, they are held together. They are held together with a bond called a hydrogen bond, and it's fairly weak. So it is a weak hydrogen bond holding these two things together. You might be asking, why would you want a weak bond? If you're going to be breaking something apart and you need to use very little energy to be able to open it up and read it, like for example, if you have a book with a bunch of information in it, you don't want it to be made of some kind of material that would like you don't want the pages glued together. It makes no sense. You want to be able to open it up, get the knowledge, and get out. You don't want to have to really try a lot just to open it up. It wastes energy, it wastes time. So the weak hydrogen bond will hold these two together pretty securely, but they're easy enough to pop apart when we need to read the DNA. This is how you will see the DNA drawn most of the time. You could also probably draw it like this. Phosphate, sugar, nitrogen base. That's one, remember? That's one little section. Phosphate, sugar, the sides are connected. And then on the other side, you have the exact same thing. Phosphate, sugar, nitrogen base, and so forth. All of them held together in the middle by our weak hydrogen bond. So let's say this was our A, our adenine, or A, adenine, it's going to bond with our T. What do we have? Purple and orange in the other ones. C is going to bond with G. So that means G and C. They're interchangeable, either way. Phosphate, sugars, nitrogen bases. This is just a combination of letters. So my body wants to determine what eye color I have. Or let's make something simple, because your eye color has already been determined. It needs the instructions. You get a scratch. It needs the instructions on how to make skin. So it's like, okay, in our blueprint, we have how to make skin. In every single cell, doesn't matter. It can be a liver cell. It can be a cardiac cell, like in your heart. It can be a neuron, brain cell. It doesn't matter. They all have your entire DNA sequence in them. This does not mean that they will use the entire DNA sequence. This simply means that inside those cells, they have your entire genetic code. This cell may only need 0.01% of that DNA. It may never use all the other pieces. However, it's basically like saying, okay, I'm going to build a giant apartment complex. Here's the blueprints for the entire apartment complex. That includes the rooms, the bathrooms, the maybe like a cafeteria, some vending machines, parking lot, parking garage, everything. I have this all on one piece of paper. So a guy comes in and says, he's like, okay, I'm here to install or to build all of the bathrooms and all the apartment complexes. So you give him the entire blueprint. He doesn't read the other stuff. He's only looking at what he needs. He's only looking at the specific section that talks about plumbing and stuff for the bathroom. An electrician comes in and needs to run the wires. So you give him the blueprints that says, here's where you're going to run all the wires. He's not looking at the bathroom and the toilet placement, even though it's on the blueprint. Every single cell gets the full blueprint, which is roughly 6 feet of DNA, which is kind of insane considering every single flake of skin, every single skin cell you have has a nucleus, and then 6 feet of this DNA jam-packed in there. This is another reason you have the double helix and the twisted ladder shape. It is very efficient at squeezing it and coiling it and packing it in. Here's our guy's DNA that we already have. We're reading this section. if we read his DNA right here, because we're going to read along one line. So we're going to read this DNA. His DNA is ACG on the left side. That is what is over here in pink. That is one section of DNA. You can have 6 feet of this stuff, and this could be, again, one tiny little bitty millimeter of it. ACG is going to tell our body to do something. Our body reads ACG, and it says, oh, that's the blueprint, that's the code for blonde hair. So it's going to give you blonde hair. On the right side, TGC might be, oh, that's the code for blue eyes. So it's going to give you blue eyes. If this had been different, like for example, if this first one was an A, it was C, and this first one, since A doesn't bond with G, C and G bond together. So basically we replace this and we replace this. This is a different sequence. CCG might not code for brown or for blonde hair. It might code for brown. GGC might not code for blue eyes. It might code for brown eyes. Again, this is a very simplified version of DNA. You have to contend with 6 feet of this stuff. Every single section can be strings and strings and strings and strings of it, and it can have a huge amount of letters. It's not just 3. It could be 30. Anything. The string of letters is what actually determines it. If one thing gets changed, it's a different sentence. You can have a sentence of 10 words. If you replace one word in the middle, it might change the entire meaning of the sentence. So this little section over here, say this is our 3 section that we just did, and it coded for our, let's say this is the right side. So GGC is over here. These are the 2 same things. GGC coded for brown eyes. So this section that tells me what to do for a very specific trait. A trait is something you have that is like a kind of a distinguishing characteristic. Blue eyes, eye color, skin color, height, weight, earlobe, attachment. All these things that are different and are passed genetically, these are traits. This little thing containing a trait, or containing basically the section of DNA that tells you what to do for that thing, it's called an allele. You can call it just a gene. A lot of times people will say, okay, it's in my hair color is from my dad, it's in my genes. That means that your dad had a very specific section that said, this is going to be the color of your hair. You inherited that from your dad, so your genes are his genes. You don't have all of your father's genes, you don't have all of your mother's genes. You're gonna get a combination of the 2 when you're created. So you will look sometimes a lot like your father, a lot like your mother, maybe a little of both. It just kind of depends where you got your genes. You may have gotten a bunch of traits that your mom doesn't really show from her father. And you'd be like, hey, why do you guys not have the same traits I did? It doesn't mean you're adopted, so don't go freaking out thinking you're adopted. It just means you got it from somebody else in your line. Alright, here we go. Super review time. DNA. The shape is a double helix. Inside the DNA, we have a monomer, which is one section. This is going to contain a phosphate, a sugar, and a nitrogen base. The nitrogen bases: apples in trees, cars in garages. A's are going to bond with T, C is going to bond with G. A section of DNA. This is the genetic code. This is going to tell you a specific trait. It's going to code for an allele. It's going to code for something about you. Maybe this codes for blue eyes. DNA stands for deoxyribonucleic acid. We know there are 2 nucleic acids from our molecules of life, DNA and RNA. DNA, deoxyribo. This is the sugar. This is the only difference really between RNA and DNA from a kind of molecular or chemical standpoint. Here's our sugar over here, deoxyribose. If this was RNA, we'd have a ribose, ribonucleic acid. We won't worry about RNA too much right now, but it's another type of genetic material. And that about sums up the basics of your DNA. You got the polymer over here. Polymer is the whole string of DNA. And you have the monomer right here. A nucleotide. DNA does look complicated when you first look at it, and everybody always tends to panic when they see stuff like this. But remember, we're looking at building blocks, we're looking at tiny things, we're clicking Legos together, we are trying to build bigger and bigger things. So as long as we understand the tiny things when we start, as we build and this gets more complicated, it becomes so much easier. And believe me, it took scientists years and years and years to even map this stuff, which means to even figure out what the human genome and the human gene sequence is. So if it takes you, couple weeks to really grasp this, I think you're doing okay.